चलो ठीक है। तो ये जो स्टार्च का सिंथेसिस होता है, उसमें आपने देखा कि 18 एटीपी एंड 12 एटीपी इट्स रिक्वायर्ड। तो आउट ऑफ these 6 ATP, they are exclusively required for regeneration of RUBP. Okay, and the NADPH, some for major for regeneration and few for synthesis of starch. Then the starch which is synthesized in the leaves, from there some of it is translocated towards the economically important parts, either the grain, the tubers, or wherever it has to be stored, or some of it may be used for maintenance, growth, maintenance, development of other plant parts like stem, root, etc., and immature leaves too. So there is partitioning of photosynthase. This partitioning of photosynthase means if the photosynthesis which takes place in the leaf, from there it is translocated towards the economical important part sufficient amount, then only the yield will be good. Okay, so this starch biosynthesis, it takes place in chloroplast during daytime only, and from RUBP, this you haven't to study, so I will not be dealing. Amylose, amylopectin are formed and then they form starch. ठीक है? So in potato, wheat, rice, where this starch is the stored food material, it is synthesized in chloroplast. And then the sucrose, basically in sugarcane, the food material, this is the stored food material. It takes place in its synthesis takes place in cytoplasm. Sucrose is made up of one molecule of glucose and fructose each. ठीक है? So this sucrose is synthesized in cytoplasm, and there the food material in sugarcane, the food material is stored in the form of sucrose. जो मेजर शुगर होता है शुगर केन में, वो क्या होता है? सुक्रोज होता है ना? हेलो?
यस मैम।
हाँ, तो वो वहां पर होता है साइटोप्लाज्म में। स्टार्च कहाँ होता है? क्लोरोप्लास्ट में। ठीक है, तो ये था C3 पाथवे इन C3 प्लांट्स। तो ये जो पाथवे है बहुत लंबे समय तक इट वाज थॉट दैट दिस इज द ओनली photosynthetic pathway which occurs in higher plants and also in algae. But later on, in 1960, these scientists you may note down Hart, HRTT, Bird, and Koschuk. So these three scientists in 1965 They reported that a 4 carbon containing dicarboxylic acid named malate and aspartate were the major products, labeled projects. In fact, we can say because carbon 14 was taken, radioisotope of carbon CO2, it was taken. ठीक है. So these two compounds, and it was observed in sugarcane leaves. Okay. So these findings, from these findings, it was concluded that in addition to the C3 pathway, yet another pathway, the C4 pathway, named it as C4 cycle or C4 pathway, is observed in the plants. And this was confirmed by Hatch and Slack in 1966, again in 1970 also. Finally, it was confirmed. So previously, only two products, four carbon containing compound dicarboxylic acids, only two products were found by the three scientists in 1965. That was malate and aspartate. But in this year, in 1966 to 70, they Hatch they reported that in addition to malate and aspartate, one more four carbon compound, oxaloacetic acid, was also observed when sugarcane leaves in sugarcane leaves itself, when they were exposed to labeled CO2 for a very few seconds, for 1 second only. Label CO2 for 1 second. But when the leaves were exposed to longer periods of time, what they reported that instead of four carbon compound, three carbon compound, 3-phosphoglyceric acid was observed, and many hexose, monophosphate, sucrose, etc. were observed. Okay, so finally it was concluded by Hatch and Slack that another CO2 fixation pathway has क्या है C4 डाइफो amaranthus. These also this C4 pathway operates. Okay, now these C4 plants, they have some mechanism which is different from the C3 plants. These plants can be distinguished from C3 plants. First is that in these C4 plants, an important physiological phenomenon, photorespiration, is absent. We'll see just immediately or in the next class. So this photorespiration is absent in C4 plants. And secondly, there are some anatomical dissimilarities, differences between C3 and C4 pathway plants. And these anatomical differences are in C4 plants, the bundle sheath cells, it's in C4 plants, the bundle sheath cells, they are surrounded by tightly packed parenchymatous cells, which are known as, so what I told vascular bundles, na? These vascular bundles, they are surrounded by tightly arranged or compactly arranged parenchymatous cells, which are known as bundle sheath cells. Now these bundle sheath cells in turn are surrounded by many, this is generally one layer, but the other layer, which is again parenchymatous cells, but they are loosely arranged into three layers, and these cells are known as mesophyll cells. But these mesophyll cells, they are absent in C3 plants. And both these bundle sheath cells and mesophyll cells, they contain chloroplast. Now you can see here that these bundle sheath cells, okay, these bundle sheath cells, they are radially arranged around the vascular bundles, sorry. And it is in a ring-like manner, isn't it? So, because these bundle sheath cells are arranged around the vascular bundles in a ring-like manner, the anatomy of these plants is called Kranz anatomy, Krantz. And this Kranz, it's a German word, and its meaning is reed-like. Reed means ring-like. Reed or ring-like means the vascular bundle sheath cells are arranged in a ring-like manner around the vascular bundles. Okay. Now, I have already said that these bundle sheath cells as well as mesophyll cells, both of them, they contain chloroplasts. Okay. हेलो हेलो
यस मैम।
हाँ ठीक है खाली सीआर बोलेगा ये चीज मैंने देखा है कि आइदर ओनली वन स्टूडेंट विल टेल यस समझ, मैं जब रुक जाती हूँ लिख लेते हो तो फिर कुछ तो बीच में बोला करो, ठीक है। नाउ एज आई सेड कि बोथ द bundle sheet cells and the mesophyll cells, they contain they contain chloroplast, but there are differences in the chloroplast of these. Okay, these bundle sheet cells, as I drew in the figure, यहाँ से they are parenchyma cells. Okay, but the shape it was cylindrical, ठीक है, and there I had drawn small ये circular, cylindrical, CYLI. बंडल शीट सेल्स में सिलेंड्रिकल सेल्स होते हैं, ओके, दे आर थिक वॉल्ड, these are very important points which you should know. They are larger, lack grana, okay. So this is the case with bundle sheet cells. Unlike these, in the mesophyll cells, the cells are not cylindrical, they are circular type, oval type. And just opposite it will be thin walled, smaller, and presence of grana, or they contain grana. Okay? Hello?
Yes, ma'am.
Yes, ma'am.
Yes, ma'am.
ठीक है, तो क्रैंस का मतलब होता है रिंग लाइक और रीड लाइक. इट मीन्स द bundle sheet cells are surrounded around the vascular bundles in a ring like manner. It may be 1 to 2 layers also, ठीक है, and this in turn are surrounded by mesophyll cells. Okay, up to here.
Yes, ma'am.
So this bundle sheet cells, they lack grana. So there will be no photosystem 2. Will there be? PS2 कहाँ होता है? Thylakoids में ना, stacked layers of the grana, है ना? हेलो,
Yes, ma'am.
हाँ, ठीक है. तो जब PS2 फोटोसिस्टम 2 नहीं होगा तो क्या चीज नहीं बनेगा?
NADPH2.
NADPH नहीं बनेगा. ओके, गुड. तो जब NADPH नहीं बनेगा तो these will be dependent on mesophyll cells for NADPH. Chloroplast of mesophyll cells, I will mean to say. ठीक है. On the other hand, mesophyll cells, they have grana, so they will have photosystem 2, and so NADPH will be formed there. फोटो सिस्टम 2 जब नहीं है प्रेजेंट, रहना नहीं है, तो कौन सा पिगमेंट सिस्टम एब्सेंट होगा, फोटो सिस्टम एब्सेंट होगा? साइक्लिक या नॉन साइक्लिक?
Non-cyclic.
Non-cyclic.
नॉन साइक्लिक will be absent in bundle sheet cells. ओके, और हमने ऑलरेडी C3 साइकिल पढ़ रखा है। ओके, तो बंडल शीट सेल्स में रूबिस्को प्रेजेंट होगा या एब्सेंट होगा?
प्रेजेंट होगा मैम।
रूबिस्को है, प्रेजेंट होगा। ठीक है, अभी हमने C4 साइकिल नहीं देखा है, लेकिन क्लास में अगर पहले पढ़े हो तो थोड़ा सा यहाँ पे बोल देती हूँ कि बंडल शीट सेल्स में रूबिस्को के अलावा मैलिक एंजाइम भी प्रेजेंट होंगे। ठीक है, लेकिन मेसोफिल सेल्स में पेप कार्बोक्सिलेज एंजाइम विल बी एक्टिव। ठीक है, so these were the major differences between the two types of chloroplast of the two types of cells of chloroplast, two different types of chloroplast. Okay, up to here. अब मेन मैकेनिज्म फॉर C4 पाथवे, आप लोग उसको खोलो। Cycle I have sent this one 5 minutes before I had sent, अगर आप लोगों ने देखा होगा तो। अभी तक आप लोगों ने जो पढ़ा है उसमें जो पाथवे पढ़ा था, ड्रॉ करते भी होगे, तो सिर्फ एक ही पाथवे जिसमें एक ही C4 कार्बन C4 कंपाउंड, कौन सा पढ़े हो पाथवे? मैंने 1 2 3 लिखा है, कौन वाला आप लोग पढ़े हो? हेलो, आई थिंक सेकंड वन क्या? हेलो,
Yes, ma'am.
आई
Yes,
थिंक
ma'am.
सेकंड वन यू हैव स्टडीड टिल नाउ इन योर 12th क्लास। हाँ, एक चीज और बताना चाहूंगी, जो C4 पाथवे था आप लोगों ने 12th के पहले भी पढ़ा होगा, उसमें कार्बोक्सिलेशन, रिडक्शन एंड रीजनरेशन फेज तीनों पढ़े हो। ठीक है, लेकिन सिर्फ लिख देते हो कि RUBP से 3 फॉस्फोग्लिसरिक एसिड बन रहा है, 3 फॉस्फोग्लिसरिक एसिड से 3 फॉस्फोग्लिसरल्डिहाइड बन रहा है, 3 फॉस्फोग्लिसरल्डिहाइड से डायरेक्ट RUBP। अगर सिर्फ इतना ही बना के साइकिल रखोगे, एग्जाम में लड्डू मार्क्स आएंगे। ठीक है, तो उसके जो बीच में के इंटरमीडिएट प्रोडक्ट्स हैं, आई हैव सेंट यू द फिगर ऑफ दैट। उसका जितना भी बीच का इंटरमीडिएट प्रोडक्ट्स बन रहा है, जो 3 फॉस्फोग्लिसरल्डिहाइड के 10 मॉलिक्यूल से 2 फ्रॉम ईच THAP 3 PGLD, तो ये डिटेल पाथवे आपको बनाना है विद देयर एंजाइम्स, विद द नेम ऑफ एंजाइम्स, कंप्लीट साइकिल बनाओगे। ठीक है, यही अंतर है जो 12th में आप लोग फोटोसिंथेसिस पढ़ते थे, जो अब पढ़ रहा हो। और बहुत सारे बीच बीच में बता रहे हैं थोड़ा हायर लेवल, लेकिन जनरली मैंने अभी तक के बैचेस को देखा है, मैक्सिमम मार्क्स वो लोग वहीं पे लूज करते हैं। ठीक है, जो 12th में पढ़े हो उससे आगे ऊपर उठते ही नहीं हो। तो हर बैच में यहीं पर मार्क्स जो लूज करते हैं। कहने के लिए कह दे तो बहुत आसान है कि ये पढ़ा हुआ है, कुछ नया चीज नहीं है पढ़ने का, एग्रोनॉमी पैथोलॉजी ये सब नया है, बट इसमें बहुत सारा नया चीज है, आप लोग नहीं पढ़ते हो इसलिए मार्क्स कम हो जाता है। एग्रोनॉमी पढ़ते हो, पैथोलॉजी मतलब कहने का मतलब है फिजियोलॉजी छोड़ करके बाकी सब डिसिप्लिन पढ़ते हो, इसलिए उसमें मार्क्स आ जाता है। तो यहाँ मार्क्स नहीं आने का सबसे बेसिक रीजन यही है कि तुम लोग जो हायर लेवल पे नहीं पढ़े उठते हो। ठीक है, तो ये चीज प्लीज ध्यान में रखना आगे। एमएससी में भी है फोटोसिंथेसिस, एमएससी में भी हम पढ़ाते हैं फोटोसिंथेसिस, लेकिन इससे थोड़ा सा ऊपर और उठ के। समझ गए? हेलो।
यस मैम।
हाँ, तो देखिए क्या होता है, फिर उस पर वही अटक जाएंगे लोग कि क्या करेंगे। तो अलग बात है, अदरवाइज 12th में मैंने देखा है कि उतना ही स्टार्ट। ठीक है, तो ये जो इसमें देख रहे हो आप लोग, सबसे पहले क्या होता है, दिस CO2, ये जो CO2 है, this CO2 it is diffusing, इसमें there is spatial separation. The initial carbon fixation is occurring in mesophyll cell, and then the second one in bundle. Just hold for a minute. हेलो, हेलो।
Yes, ma'am.
हाँ,
यस
तो
मैम।
यहाँ पर इनिशियल जो CO2 फिक्सेशन है, इट इज इन mesophyll cells and again in the bundle sheath cells it is refixed. तो ये जो mesophyll cells में है, this carbon dioxide will diffuse inside the chloroplast of mesophyll cells, and then this enzyme, very important one, carbonic anhydrase, this will convert this CO2 into bicarbonate ion. Okay, and this bicarbonate ion is not used as such during photosynthesis, but this bicarbonate ion, it
Excuse me,
is
ma'am.
again converted into CO2. Okay, when
Ma'am, your screen
photosynthesis,
is not visible.
मैम इसकी मैम
oh,
स्क्रीन शेयर कर
so
दीजिए प्लीज।
sorry. हाँ, तो ये जो कार्बोनिक एनहाइड्रेज, इट इज एन इम्पोर्टेंट एंजाइम। ठीक है, what it does, it converts CO2 into bicarbonate ion. ठीक है, And this bicarbonate ion is not used as such during photosynthesis in C4 pathway, but will again be converted into CO2. So this reaction is in a is bi it will be bidirectional. Okay, here conversion, here only it will again convert into CO2. Okay, when photosynthesis is to take place. चलो ठीक है. So the first product of carbon fixation here is oxaloacetic acid. Okay, first product of carbon fixation here is oxaloacetic acid. Now this oxaloacetic acid, there are three pathways. I have written 1, 2, 3. Okay, so this oxaloacetic acid is converted into malate. This malate, it will diffuse from mesophyll cells to bundle seed cells. Again, this will be converted to pyruvate plus CO2, and then this will be refixed through Kelvin cycle. Chance देखना पड़ेगा आपको. हम तो भाई क्लास में पहले आप लोग फोन करके लिए आते हैं. चलिए पहले फोन करके आना चाहिए. Got it up to here? So this CO2 is refixed through Kelvin cycle, C3 cycle in bundle sheath cells. Okay, up to here? Hello?
Yes, ma'am.
Now, the other pathway, one point about this, that this occurs exclusively, mainly in maize and sugarcane. You may note it. This pathway, it occurs in maize and sugarcane. Now this pathway, it uses NADP. Okay? And another, this malic enzyme as well as RUBP is Rubisco enzyme is active. Okay? बॉटल में पानी भर लिया. दूसरी बॉटल में पानी भरो, नहीं तो नीचे उतारते ही जाएंगे. बाल्टी में भर लो. Okay, up to here?
Yes, ma'am.
So because this it uses NADP as well as malic enzyme, so it is also known as NADP ME type. The other pathway, second one, this one. So in this pathway, the malate or the malic acid was converted directly into pyruvate. But in the second pathway, this oxaloacetic acid, it's converted first into aspartate in the bundle sheath cell itself, and this aspartate in the bundle sheath cells, this will then diffuse to mesophyll cells. This will then diffuse to bundle sheath cell here, and then through transamination again it will be converted to oxaloacetic acid, and then it will be again converted to pyruvate and CO2. Okay, now this CO2 is fixed here, refixed. So this is the second fixation here. So here you can see one important enzyme, PEP carboxylase. ठीक है? So it is PCK type. And it takes place in Panicum maxicum and Chloris guyana. Okay? Panicum maxicum I had already written in the beginning of the spelling of Panicum, I think. Okay, up to here. Now, the third pathway, this oxaloacetic acid, just like the second pathway, is transaminated into aspartate in the mesophyll cells, and from there it diffuses into the bundle sheath cells, again transaminated into oxaloacetic acid, and then it is converted into malate. The first pathway, the malate. Okay, now here it requires an important enzyme, malate dehydrogenase, and this, in addition to an enzyme, it also needs NADPH. Sorry, NADH. So this malate dehydrogenase, it is dependent, because it is dependent on NADH, so it is NADH dependent malate dehydrogenase. Okay, now this malate, it's again converted to pyruvate plus CO2. This CO2 is refixed. Okay, because it requires NADH, so this is also known as NAD as well as malic enzyme, NAD-ME type of carboxylation. Now this occurs in Atriplex spongiosa, Portulaca oleracea, Amaranthus edulis. Amaranthus का तो नाम सुने होंगे. I am writing only the species. Edulis. ठीक है, Atriplex spongiosa. स्पेलिंग लिख लोगे, गलत मत करना. Hello?
Yes,
Hello?
Yes, ma'am.
Okay, up to here?
Yes,
Okay, up to here? Time हो गया है. So what is the biological significance of this cycle? That plants belonging to tropical areas in Zari conditions also where there is low water content, high temperature, bright sunlight, and there is closure of stomata, this will not affect the photosynthesis, and it will occur even when there is very low concentration of CO2. ठीक है? As I've already said that here the photorespiration is absent, so photosynthetic rate will be higher. It means it will be twice as efficient here in converting the solar radiation into production and in the production of dry matter. ठीक है? किसी ने अटेंडेंस लिया है? स्क्रीनशॉट जिसमें रोल नंबर आता हो जिसका?
मैम अभी ले लेते हैं।
हाँ, रोल नंबर भी मुझे चाहिए, उसमें रोल नंबर नहीं दिखता है जनरली।